This is a production of Cornell University.
Thank you all for coming and for this opportunity to talk to you about what we do in Geneva at the PGRU. I have quite a bit of material in my talk. I try to touch on a lot of different things because there are a lot of different things that we do, and so I try to tell you a little bit about everything. So I'll talk about the National Plant Germplasm System and PGRU and what we do at PGRU. I'll also talk a little bit about the GRIN Global Database where you can order germplasm. I get a lot of questions about that. And I'll talk a little bit about the molecular research that we've done and also about our outreach activities. So the NPGS, it's a cooperative effort by the state, federal, and other organizations to preserve genetic diversity. And the idea is that genetic uniformity increases crop vulnerability to pests and stress and disease. So NPGS provides access to genetic diversity so that breeders, researchers can bring forth new varieties that can tolerate or resist these diseases and stresses. So we serve science and education. We acquire, preserve, evaluate, document, and distribute plant germplasm. And this is just to show you, it is a national system and it's spread throughout different growing regions so that, for example, the crops that we have in Geneva, New York are things that do well in our temperate climate. As far as NPGS, as of October 2017, there were over 15,000 species conserved and over 583,000, 500,000 accessions. And an accession you can think of as a population. It's a genetically unique stock or population or clone of a tree that is conserved as an entity. So as far as samples of seed or clones distributed, around 250,000 per year throughout the whole system, and about 30% of those are international. And that's just to show you that there are 29 repositories, seed crops, clonal crops, genetic stocks, and where some of the larger repositories and collections are. And that on the that list on the side is from the GRIN Global database, if anybody ever wanted to look that up. So as far as in Geneva, the seed crops repository, we hold around 200 taxa and over 12,000 accessions. And our major seed crops are listed here: onion, celery, different brassicas, winter squash, buckwheat, tomatoes, our largest collection, and radish. In 2016, we distributed close to 8,000 samples of seed and 254 orders. And so I'll talk about some of the activities. When we acquire germplasm, the purpose is to fill gaps in our collections. If people go out on collecting trips, those are administered by the Plant Exchange Office in Maryland, and they do administer domestic and foreign plant explorations, and they review proposals, and I usually review some of these proposals every year and rank them and to support ex situ and in situ conservation of crop plants and wild relatives. For example, for PGRU, there was a collection in 2002 of tomatillo, and the former curator, Dr. Robertson, and Dr. Todd Vision from the University of North Carolina did a joint collection, and they collected 111 accessions, including some large fruited types that we had never had before, and some self-compatible types that could be inbred, which is unusual for tomatillo. In 2013, Dr. Zong, who's also the research leader at PGRU, and Dr. Robertson made a trip to the Chinese Academy of Agricultural Sciences in Beijing, and they did some germplasm exchange. And I was telling Don I was supposed to fly to China tomorrow for a similar opportunity, but because Trump is in China this week, I can't go. Hopefully it'll happen at the end of the month, next month. So for our regeneration, it's quite a big project. We do a couple hundred plots, usually on average 150 to 200 plots per year. So each plot is a population or accession, and we cover them with these nylon mesh cages when they're ready to flower, and then honeybees are introduced. We rent the honeybees hives, and we usually do about 8 different species or taxa per year, including biennials and annuals. And we have different size cages depending on the crops. The winter squash go into the largest cages. They take up a lot of space. So this is just some pictures of the cages. They have to be isolated from each other. So we use a plow to put soil over the edges, and then they have zippers where you can go in and out of the cage. And we use black plastic to grow everything except for the tomato, and then there's drip tape underneath the plastic. For the biennials, they have to be overwintered. So for example, all the cabbage are dug out of the field. They're ready to do that now, but it rained so much, we're not sure when we're going to be able to dig them. But they go into these wooden crates with sawdust, and then when they're planted, you can see they're just stubs in the field. They take off all the heads, and they got that that meristem, that growing tissue tip there, and then those are going to start. And then here you can see when they're in the flowering stage, there's a cage around it. Most people don't see cabbage flowers because you eat the head and that's it. But that's a second year what happens. And then some things, they might not flower, they might not have enough time, so we dig them up, like these collards, the picture of the collards. They were brought in, fertilized for like 6 or 8 weeks in a cold chamber in December, and then in January, they flower in the greenhouse, and those are hand pollinated. So depending on the crop, you have to sort of custom regenerate it. And then we dry the material in mesh bags that are hanging out at the barn. And then we have different types of machines to thresh the seed depending on the crop. And then for the seed storage, the seed will come in from the barn and it will go into this thing called the ante room. So the pre-ante room is where we do like seed orders. It's controlled humidity and temperature. You know, we weigh the seed in there. Then the ante room, it's more like a refrigerator. And let me see, there's my pointer. That's the ante room here. So we dry down the seed in that room to 6% relative humidity, and then it's stored long-term. And then this is the long-term storage facility here. So it's basically a freezer. We do characterize, the time to characterize for us is when we're regenerating, so we can only do highly heritable traits. We do collaborations with breeders and other scientists. If we get some grant funding, we can do more traits other than just highly heritable traits. We can do things in replicate. For example, in 2015, we did a field trial of our Brassica rapa accessions in collaboration with Michael Gore and Chris Pierce. This year, we started a new way to collect data. We bought this small Android tablet. It's very lightweight, easy, and this Fieldbook freeware, and it's spreadsheet compatible, so we can collect data in a way that we can easily upload it into the Green Global database. And I've been talking with Steph Gaylor at the Long Island Regional Seed Consortium about, they are developing a smartphone app that they'll share with us. So maybe, hopefully it'll make it easier for people that receive our material to collect data for us. That's what they're aiming for. And we did collect data on 6 different crops this summer. And this is just a screenshot of the field book giving the list of traits. So the person that put this in, she put AL, that's the crop, so she put that as a prefix to every trait, so all the traits would sort together by crop because they're alphabetical. So AL in this case is allium, and then BB is biennial brassica. And here's another screenshot. So it's a very nice interface. It's a very big interface. So this is plot 410. Lyco is Lycopersicum, is tomato. The trait is leaf type. And then you can see the definitions are right there: dwarf, potato leaf, etc. So she can just type in the number 3 and then hit this one, and it will scroll to the next plot. And then you can also use a photo as a trait. It will collect the photo and it will label the photo for you with your plant ID, which is nice. So we get images of plots, of plants, of head shapes as our traits. And those go into the Green Global database. We do also scanning of fruit. And this year we tried to scan radish, but it didn't scan very well because it's so dark. So she ended up using a camera. And they don't look that nice. I mean, you have to remember they were grown for seed. They weren't really grown for the radish, the edible portion. But we've to give people pictures is better than to not give them pictures. So we think it's valuable to do these images. And you can see there's a lot of diversity in our collection. So I'm going to talk a little bit about Green Global. Like I said, I do get quite a few questions about that. And it did change recently. So this is, if you just Googled Green Global and PGS, you would get, this is the starting page that you would search from. So if I suggest that if you decide you're pretty sure you're going to order something, the first thing to do, or maybe the second thing after you found what you want, but you want to register with an account and then read the help section. It's really well written. It's a good resource. So you can search for things like descriptors. So in this case, the descriptors are all crop specific. So I don't know how easy it is for you to read the screen, but this is when you go to the descriptors link, you get the list of crops. I picked maize, and then you get the traits for maize coming up. So I picked kernel color, you put a check mark, and then you could say equal to, then I said red. And then you do the search, and I ended up with 802 maize accessions with kernel color red. And then you would go through and check the boxes of the ones that you want to order. And if you click on the name of it, you'll get a more detailed page. And I'll show you that in a minute. There's another way to order. If you had a list of accessions, like PI numbers usually from a paper or something, the search box expands if you say using a list of accessions, and then you can just copy it from a spreadsheet and paste it in here and get the search that way. Okay, this is what an accession page looks like. If you click on the accession, this is one PI of tomatillo. You see the image. And there's also, if it has it, it will have map data in there. It also has information here about its source and things like that. So if you click on that map for this one, the green dot is that one accession, and then these other dots are other accessions, and it's colored by number of accessions. So yellow is one accession and more accessions. So if you were trying to do a geographical survey or study, this would help you to pick your material possibly. You can also use multiple criteria. You do advanced search, and then in this case, you know, there's a drop-down list of things you can pick. I picked here country of origin, Brazil, the state of Rio de Janeiro, and then, for example, you could say improvement level is breeding material, and you can go on and add multiple criteria. One thing I really like is the GRIN taxonomy. That's a really good resource for anybody working with plants. It gives the common names, it gives conspecific taxa, and it gives synonyms. So if you're reading an old paper and it's a genus and species and you want to see what the current name is, it's a really good way to do that. Or common names, you know, and you're trying to look up the common name. It's a very scholarly set of information, and it's very well curated. So there's a taxonomy link there. Okay, so now I'm going to switch to talk a little bit about some of the genetic research that has gone on in my lab. We've done quite a bit of genotyping of collections, and the method that we've been using is genotyping by sequencing, and you're probably familiar with it because a lot of that was developed here at Cornell. It's fairly inexpensive, $20 per DNA sample if you do 96 well format set of samples. And because you're finding your mutations, your markers, or your SNPs at the same time that you're genotyping them, you don't get the bias, whereas if you just had a set of markers and you use that set to genotype your material, you wouldn't find the additional markers. And it doesn't require a reference genome, and that's really important for us because with the exception of, you know, tomato and Nebraska rapa had a good reference genome, but some of the other crops don't have any reference genome. So this is that same slide I showed you earlier, our major collections. And what I did was I just put an asterisk next to the ones that we've done some GBS on. So we've done most of the major collections, and I can't say we've done all the accessions, but we've done sampled accessions, and we pick the samples based on certain criteria such as geography or morphology. So I'm going to go through these just briefly to tell you the highlights of these different studies. So the first one I'm talking about is Ayan, Allium cepa. It's highly consumed in the United States. Its economic value ranks third amongst the vegetables. It's an ancient, globally widespread crop, and there's thousands of onion cultivars that have been developed to fit very diverse environments and dietary preferences. It is a costly crop to breed and conserve because it is biennial. It requires cross-pollination. It's very day length sensitive as far as whether or not it will flower. Also, it has a very large genome, and so it's very hard to develop molecular genetic markers that breeders could use. When we did GBS, we found 70,000 markers, single nucleotide polymorphism, SNP markers, and we used doubled haploid lines as controls. Those doubled haploid lines should be homozygous. So whenever a marker was not homozygous in one of those DH lines, we threw it out. And we also had a mapping population, F2 mapping population, that our collaborator, Mike Havey, had developed. So we also used the expected segregation ratios in that population to decide whether markers were high quality markers. So doing this, we started with 70,000, we ended up with 900 markers. But those 900 markers should be good high quality markers. So we concluded, and we're gonna write up a paper. What I have at the bottom here is just a poster. That GBS is promising. I still have to analyze the data. We have 95 of our accessions, also genotyped. But I'll say it is promising, but we had to, Jeff Globitz, who works here at Cornell, had to make custom scripts to analyze the data because it was so challenging with this large genome. And I tried to analyze it multiple times, and I just didn't get very good results. So with his custom work and expertise, we were able to do it. The next one I'll talk about is Nebraska rapa. It's different types of Asian greens. It can be consumed raw or cooked. It's you might know it as Mizuna, Chinese cabbage, bok choy, spice, sarsen. It's also root vegetable turnip, which is also used as animal fodder, and it's also used as a cooking oil. So these are all different morphotypes that have been developed, across the globe, but the genetic relationships haven't been well understood. And if we knew more about how they were related to each other, that would help breeders and also conservation. So we were able to sample Brassica rapa from our gene bank and also from the gene bank in Ames, Iowa. And we genotyped 10 different morphotypes. And we ended up with 333 accessions by over 18,000 SNPs. And this was published this year earlier. Kevin Bird was a student in Chris Pierce's lab. And he took the lead on this and published it. And what he found was that there was a genetic distinction among the morphotypes, but also the same morphotype might be genetically different if it had a different geographic origin. And so that was the largest data set of this kind to date. Next one is tomatillo. It originated in central Mexico. It's widely cultivated in Guatemala and Mexico. It's primarily conserved as relishes or sauces, such as green salsa. And it's becoming increasingly important in the USA as a niche crop. It is considered an underutilized species by the FAO, and breeders might be interested in size, disease resistance, and mechanical harvest. Previous studies of populations showed them to be closely related to each other. That was work by Todd Vision, who also went on that collection trip with Larry. So we GBS genotyped 125 accessions, and we got a pretty large data set. And basically, to summarize, we did find a lack of relationship based on geographical origin, similar to other researchers have found. But we did identify 86 pairs of accessions that were quantitatively different from each other. And we were able to graphically represent qualitative divergence among the 125 accessions. And we also confirmed the homozygosity of some new inbred lines that Larry developed. The next one I'll talk about is wild tomato. So this one very diverse wild tomato that's used a lot in breeding, Solanum peruvianum, it was recently reclassified into 4 distinct species, but there is gene flow among the species. So we genotyped 46 plants representing different species, about 12 per species, and we used 14,000 of the markers. And we did find that based on geography, isolation by distance is the geographical distance. So that means we found support for isolation by distance, which means that things that were sampled in more remote distances from each other were more genetically different from each other. And especially this pattern was observed if it was between the pairs of species. So that supported some idea that there is reproductive isolation between the species. And we found 2 of these new taxa weren't significantly different from each other genetically. But we did find a lot of markers that we could possibly use as diagnostic markers to identify the species. And this is just a graph of what I just mentioned, that the genetic distance is here, and then the log of the distance in kilometers is here. And within a species, the points are red, and between species, the points are blue. So you can see there's a higher slope in the blue one, and it was significant. So that's supporting that there is more reproductive isolation by distance between species than within a species. The next one I'll talk about quickly is winter squash. It's been valued for its good storage properties. It has a thick, hard skin, and it provides a wide range of nutrients. Consumer demand has been increasing during recent decades, and it has some naturally occurring compounds that have been shown to have health benefits and also used in pest control. Improvement breeding is focused on fruit size and shape, non-bitterness, deep orange fruit color, earliness, bush habit, insect and disease resistance. And what we have are what we call core sets based on morphological diversity, 95 of our accessions, and geographical diversity, another 95 accessions from 38 countries. But these two sets overlap, so there's a total of 165 accessions. And we sampled plants, multiple plants per accession. Right now, we have the data collected, and I'm collaborating with people from QCAP, including some people from Cornell. And hopefully, they have some data too, so we're gonna put all of our data together. Radish is the next one I'll talk about. It might not be that important in the US, but across the globe, it's pretty important. It's 2% of all global vegetable production. It's consumed as a root, as a leafy vegetable in some places, as sprouts, seeds or seed pods, and it's also used as a cover crop or a forage crop. We use it as a cover crop. It's nice, it breaks up the soil, and then it provides organic material, you know, for the soil, to improve the soil. It is an ancient crop, native to Eastern Mediterranean and Eastern Asia, and there's some debate whether it's had one or multiple domestication events. And there's a lot of morphological diversity depending on where in the globe it's utilized. And it also adapts to local conditions. It's a rich source of nutrients. So we have a core set based on a weighted geographical sampling. That means we sample more accessions if we had more representation from that country. So we ended up with 35 countries, 152 accessions, and 5 plants per accession. And that data I haven't analyzed yet. That's one of the overlap interests with the people in China. They're very interested. They study radish. So hopefully our relations and collaborations with them will move this project forward. Next one is cultivated tomato. Like I said, it's our most important crop. It's our most important crop that we conserve because we have the most accessions and we get the most requests for it. It's highly consumed in fresh and processed forms. It's considered a high value but high risk crop because it is very vulnerable to diseases. Mostly you probably know that because you've tried to grow some. Mostly wild relatives are used in its breeding because it has a narrow genetic base. And it's, like I said, it's very susceptible to pests and stresses. Consumers in the US want more flavor, color. They like the heirloom types, but those require still the resistances, the shelf life, and the durability. So what we have is 185 accession core set based on geographical origins, centers by diversity, fruit shape, heirlooms, commercially important varieties. And we assayed 2 sets of 96 well plates. And when I analyzed this, we ended up with around 3,700 high quality markers in 190 plants. There's software you can use to analyze what you call the population structure. So we had Pimpinella folium is a wild tomato. We had 1 sample of that. And what this is showing, there's like 3 virtual groups, populations, the yellow, the blue, and the orange. And so what these numbers are across this axis are the 190 plants. So each of these little lines is that genotype of that one plant. So Pimpinella folium had the most yellow genome, you would call it, virtual genome, and then also some of the blue, but it didn't have any of the orange. So at this far end, genetically we have Pimpinella folium. At this far end, way up here, this sample here was a pear type, ellipsoid type tomato from Italy. It had a lot of the orange, a little blue, and a little yellow. So what you're looking for is what I point out here. There's some kind of genotypes that kind of stick out, and this is another way to look at it. It's called a neighbor joining tree. It's the same data set, it's the same data, it's just looking at it in a different way. It's more like a tree. So Pimpinella folium is out here, you would expect. So this is the most distantly related to all these other genotypes. The branch length of the tree is the genetic distance. And then what you're seeing are a lot of things that kind of just are closely related. But then you see these outliers. So these Italian types look diverged. There's this cherry tomato type here that's divergent. And then you get these other accessions that are also divergent, and they're kind of falling more towards Pimpinella folium. So what those are probably land races that might have crossed with Pimpinella folium in their history. So what we have here, this is a landrace succession that's closely related to wild tomato, yet it has this big, large red fruit. So I guess what we found is there could be some accessions in our collection that could have diverse genes, but they still have the large red round fruit, which Pimpinella folium has a small fruit that tastes terrible. So if a breeder wanted to try to get something that was divergent, but not go for that little tiny terrible fruit, they might try to use something like this. Yeah, so we're winding down here. So the other thing I just want to mention was some of our outreach and education activities. We rarely ever say no if somebody asks us to do outreach. We try to accommodate them. And one of our most popular things that we get invited for is to extract DNA from a banana using common household chemicals. And I show over here what we use. And this is an elementary school in Geneva. It's called Science Night. And you can see the kids come and they get to do it hands-on, and then they get to see the DNA. We use a toothpick, and they can pull the DNA out of the solution. They take the DNA home if they want to. A lot of them do. And they really love it. And then we give them the protocol so they can take it home and teach other people how to do it. So it's just try to educate, the public about what DNA is. Another thing that I'm pretty heavily involved in is called the Northern Organic Vegetable Improvement Collaborative, NOVIC. And people at Cornell are also involved in this, like Michael Mazurek right now. But Molly John was the one that kind of started it many years ago. It wasn't under the same name, but it's the same project that's evolved over time. The idea is to breed new varieties for organic conditions that do well in northern climates. So they try to identify the best varieties for organic farms. They work directly with farmers, educate others on organic seeds, variety trials, and on-farm breeding. And what we do mostly, because we're so good at seed cleaning, we teach people about saving seed and how to process seed, how to grow things to save the seed. So these are pictures from a fair that we do in Maine every year, where we actually bring up a trailer full of like threshers and the wet seed processor for the tomato, and we get a lot of people coming to try to learn how to do these things, and we hand out a lot of information, and we hand out seeds also. That brings people. So I'm going to wrap it up now and just thank you all for your patience and for being here. We have a big staff that I work with every day, and they make all this possible. It ranges from greenhouse to farm to the seed storage to the database person, and then Susan does research support, like the field characterization and lab characterization, and we have a lot of technical assistance. These three individuals just graduated from Hobart and William Smith colleges last spring, and then they came on to work with us temporarily and learn a lot about agriculture. And that's a picture of me and Larry Robertson, our former curator. on Meg McGrath's in Long Island tomato trials a few years ago. They have such sandy soil down there, we could use a golf cart, and that was really great. We really enjoyed that. So I'll be happy to take any questions, and thank you all.
Joanne, do you want to see if Geneva has any questions?
Yeah.
Just say Geneva.
Geneva, are you there?
Sure. Yeah.
Do you have any questions?
Joanne, that was a lovely talk. Thank you so much. I was curious with your allium diversity study, it looked like you really had to whittle down the good SNPs to what, 700 or 900? I forget the exact number.
Yeah, that's true. Yep.
Your really wacky segregation distortion that you saw in that population, and do you have any idea what the
I don't. It's a lot of it has to do with probably tandem duplications. Yeah, so like I said, I wasn't able to analyze it when I did get a set of SNPs, and then I would give them to our collaborator, Mike Havey, to map them. He found that they were clustering together. They weren't really well distributed either. So, like I said, Jeff had to really do a lot of extra custom scripts to analyze the data to get down to those 900.
Thank you so much.
Yep, thanks, Zach.
Questions here?
Yeah.
Okay, cool. I think with the increased access to sequencing information, we're seeing kind of a push to develop core sets, core collections that breeders might use in future breeding efforts. I'm curious, like, what you think from a conservation standpoint, the implications of developing such core sets, whether it's positive or negative.
Yeah, I know there's different ideas about that. Kind of, we don't want to get rid of things and say we have this core set, it represents all the diversity, we don't need the rest of the collection. That's not something that I would be in favor, or MPGS doesn't have that philosophy. It's nice to have what you call a core set. A lot of times I don't even like to call it a core set, I like to call it a diversity panel based on whatever it was developed, geography or morphology. But I imagine there is a lot of redundancy. We know there's probably a lot of redundancy, and we can't conserve all of it. It would just cost too much. But for something like tomato, the life of those seeds is 50 or 100 years the way we store them. So we can afford to keep them until somebody can sequence the whole genome. I don't think it'll be that long before it'll be affordable to sequence the whole genome of multiple plants per accession and see.
Yeah, Joanne. I want to know what makes a high quality SNP versus a low quality. How do you decide?
Yeah, that's a good
which
question.
Ones are good and which ones are bad?
Yeah, that's a good question. It goes through an analysis pipeline. So things that you look at, how many times was it observed? You have millions of sequences. So, how many times was it observed? And there's going to be a threshold. You set these parameters depending on your sample size and the biology. For example, for the mapping population, they need to be in the parents, right? We don't want anything that wasn't in the parents in that mapping population. And they had to segregate the way they were expected, and they had to be homozygous. So those are the main. The other thing is we don't want more than 2 alleles at a locus. These are mutations in the DNA, so you're not likely going to get 3 mutations. It's going to be an alignment problem with the sequence that gave you that 3rd or 4th mutation. If you have GATC, you just want 2 of those. You don't want 3 or 4 of those, because that's probably misalignment. Maybe there was an insertion or deletion nearby that caused it to look like a mutation. So those are some of the things that you set the parameters in the pipeline. We call it filtering them down.
So Joanne, you spoke about putting seeds into the deep freeze for long-term storage. Do you periodically retest the viability of that seed?
Yes. So they're tested before they go into storage. They're tested. So the things that just came out of the field in 2017, after they're given a few weeks to dry down, then they'll be viability tested. If they don't have good viability, we won't probably keep them unless it's the only thing we have. Then we would keep it. But yeah, they should undergo, depending on the crop, every 5 or 10 years. And that's the kind of thing, if we could get more feedback from people that we send it to, we wouldn't necessarily have to do that germ test ourselves. We could use their data. Like if we send somebody 50 seeds and they say, oh, they were great, we got 100% germ, we would use that, observation. So yeah, we do some of our own germ testing, and we also outsource some of it.
Other questions?
Okay, the PGRU vegetable seed collection, are there species that are self-compatible, and therefore how do you deal with that as far as maintaining?
Self-incompatible? Yeah, that's why we use the honeybees in the cages, or we do it by hand. So the accession is a diverse population. So you don't have within that accession, it's not incompatible with itself. But yeah. So we do outcross them. If we have to do it by hand with a paintbrush, we'll do it by hand. But they're going to cross to other plants in the same accession, like radish, for example. Did that answer? Yeah, we don't have like breeding line. We don't have a lot of like highly inbred material or highly inbred stocks or things, or mutant stocks or things like that. We don't generally have that kind of material. We have populations.
Okay, I want to again thank Joanne.
All right, thank you.
This has been a production of Cornell University, on the web at cornell.edu.